Ok, estamos entonces eh, en condiciones de comenzar oficialmente la sesión. Estamos grabando, al igual que ayer, para aquellas personas que no pudieron conectarse ayer al seminario. Estamos transmitiendo en vivo y obviamente la grabación tanto de ayer como la de hoy quedará en la cuenta, bueno, creo que es mi cuenta de la Universidad de Chile en YouTube, así que ahí se pueden conectar cuando quieran o ver el seminario pasado, si no pudieron estar ahí, o repetir los seminarios si es que no, algunas cosas no las pudieron entender y quieren entonces entrar un poquito más en detalle y entender un poquito mejor lo que se conversó ayer respecto a FLIM y lo que probablemente y lo que obviamente hoy día vamos a comenzar a conversar respecto a la otra parte de las metodologías asociadas a este equipamiento que va a ser instalado ahora a fin de mes. Entonces, para aquellos que no estuvieron ayer, les voy a compartir un poco esta mini presentación que hice ayer para poder entonces estar un poquito más contextualizado respecto a lo que estamos haciendo. Eh, Supongo que todos pueden ver la pantalla compartida, así que como ustedes pueden observar aquí en la presentación, estos seminarios, tanto el de ayer como el de hoy, y obviamente unos que vendrán más adelante, que esto lo vamos a confirmar, como les comentaba ayer, un poquito más en detalle respecto a los días, horas y presentadores, creo que estos días o la próxima semana, están enmarcados dentro del proyecto Fondekip que fue adjudicado hace 2 años ya y como el profesor Baúl comentaba ayer, estas cosas toman un tiempo en entrar en funcionamiento regular, sobre todo cuando son compras en universidades públicas. Pero ya el equipo está pronto a ser instalado y entonces el proyecto en el cual se enmarcan estas presentaciones tiene relación con ese equipo y que en general nosotros podemos definir como que se va a poder habilitar en la unidad de microscopía avanzada de la Facultad de Ciencias una unidad de microscopía multifotón con super resolución temporal y además con sensibilidad de molécula individual. Lo que va a permitir entonces, como sale en el título ahí, es fortalecer la colaboración multidisciplinaria como primer objetivo y evidentemente cada una de las unidades de investigación por sí sola también va a poder fortalecer su propia investigación. Entonces, eso tiene como una gran ventaja tanto como intrainvestigación como interinvestigación dentro de la facultad y obviamente dentro de otras unidades académicas. Eh, como les mostré ayer, la presentación de la doctora María Loidol fue relacionada a la visualización de células y cómo nosotros podemos utilizar la aproximación de microscopía con alta resolución temporal, lo que tiene que ver con tiempos de vida de fluoróforos, para analizar diferentes aspectos y diferentes preguntas que uno pudiera realizarse y cómo esta metodología se contrasta con la tradicional microscopía confocal, ¿cierto?, que nosotros podemos realizar en este momento en la unidad de microscopía de la facultad. Y además, metodologías asociadas a esta capacidad de obtener tiempos de vida de fluoróforos, como por ejemplo son la resonancia tipo FRED y otra metodología como la anisotropía. Entonces, lo que nos toca hoy es escuchar a la doctora María Loidolt, que nos va a hablar sobre cómo nosotros podemos además utilizar este equipamiento para enfocarnos en el estudio de moléculas individuales, es decir, cómo nosotros podemos saltar desde mediciones en ensamble, en poblaciones que nos entregan siempre un promedio, que esto nos va a conversar más en detalle la doctora, a un estudio mucho más específico de mayor resolución que tiene que ver con observar una molécula a la vez y con eso entonces poder entender diferentes comportamientos y responder diferentes preguntas también que nos podamos hacer. Por tanto, vamos a analizar en específico lo que es FRED y espectroscopía de correlación de fluorescencia a nivel de molécula individual. Y ya entrando en agosto, vamos a tener entonces un seminario respecto a cosas más específicas y prácticas del equipo, que va a estar dictado de manera presencial por la persona encargada que va a instalar el equipamiento. Creo que va a ser el 3 de agosto, voy a confirmar exactamente la fecha, pero sí va a ser esa semana. Y además vamos a tener entonces la segunda semana de agosto, 12 y 13, los días están confirmados, tengo que confirmar solamente el rango horario. charlas en las cuales investigadores que utilizan estas metodologías nos van a hablar de cómo ellos aplican tanto FLIM, FCS, FRED y FLIM-FRED para sus investigaciones, pasando de estudios de biología estructural a estudios incluso a nivel de células, utilizando todas estas metodologías. Entonces, con esto nosotros queremos generar un ciclo de webinarios o seminarios, dependiendo de cuándo lo hagamos presencial o cuándo lo hagamos vía Zoom, que le permitan a todos los investigadores o estudiantes de posgrado, en general, tener cierta idea de cómo poder aplicar estas metodologías en sus estudios en particular, y cómo estas metodologías permiten, por ejemplo, ampliar el horizonte de cosas que podemos hacer como unidad de investigación y poder empezar a colaborar en distintos aspectos. Entonces, la presentación de hoy, como les comentaba, va a estar dictada por la doctora María Loidolt, que es especialista en microscopía de la empresa PicoQuant, que es la empresa en la cual nosotros hicimos la compra del equipamiento. Entonces, ella nos va a hablar un poquito ahora de los aspectos quizás más de biología estructural asociadas a las metodologías. So, María, you are free to start your presentation. Can you share your screen? Yeah, you can share. Are you mute?
Here we go. Okay, yeah. Sorry. Yeah, hello everyone. Thanks a lot for taking the time and joining the webinar today. I'm very excited that soon you will get your LSM upgrade kit for time-resolved confocal microscopy. And therefore, I want to tell you something about the methods that you can use with this new microscope system, and also a few examples so that you can get an idea of what the system is capable of and how this could contribute to your research. So yesterday we talked about imaging methods, FLIM and FLIM-FRED and anisotropy. So today, instead, I'm going to focus on single molecule methods, on single molecule FRED and FCS. So no images today. But I still hope it's going to be very interesting for you. If you have any questions, do feel free to interrupt me. But I'm also going to take a short break. So in the first part of the webinar, I want to tell you about single molecule FRED, and then I'll take a short break for questions and discussion. And then in the second part, talk about FCS. Okay, but let's jump right in with a quick reminder about the basic process of fluorescence, since here we are talking about fluorescence microscopy. So what actually happens in fluorescence marker? Yeah, so here you can see the Jablonski diagram, and you see the one of the electrons of the molecule in the ground state. And then if the fluorescent marker absorbs a photon from your excitation laser, then the electron goes to excited state. And then after a certain time, a certain characteristic time of a few nanoseconds, the electron will relax back to the ground state and emit another photon. Yeah, and so this second photon, that's the fluorescent signal, which we are using in all of the following methods. Yeah, and so this process has certain characteristics for each fluorescent molecule that you're using. So each molecule has a certain absorption spectrum, and each one has a characteristic emission spectrum. So I'm quite sure you're familiar with this, because the excitation and emission spectra are used in all the confocal microscopy. And so, for example, for multicolor imaging, you would use like a blue, green, and red fluorophore, for example, or different fluorescent proteins that have different absorption and emission spectra to look at your molecules of interest or your process of interest. But as I said, this process of photon absorption and then fluorescence emission also takes a certain characteristic time, and that is the fluorescence lifetime. Yeah, so we excite our marker molecule with a laser pulse, and then it takes a certain short time before the fluorescence photon is emitted. Yeah, and this time can actually be measured by the setup which you will get. And if we measure this time delay very often, we will get a histogram, and this can tell us the characteristic fluorescence lifetime of the marker molecule. Yeah, and so this is just an additional dimension of information that we can now use. Yeah, so before we were using the absorption and emission spectra of fluorophores to identify them or to tell them apart. And now we are also able to use the fluorescence lifetime to get more information about what is going on in our sample. Yeah. So how does this actually work in practice? Yeah. So I have a short animation. Yeah. So here's our excitation laser, which is shooting excitation pulses into the sample, and then the sample is excited, and after a certain time, the sample emits the fluorescence photons, and some of them hit the detector, and then once the detector gets a signal, it stops these electronics. Yeah, and so these TC-SPC electronics, they are like a very fast stopwatch, basically a very fast, precise stopwatch that is measuring this time between excitation of the sample and the arrival of the photon at the detector. Yeah, and it's doing this for every photon that is detected, and this arrival time is then saved and recorded for each photon that you register from your sample. Yeah, and so this is the information that we are going to use for all the methods that I will talk about now. Okay, one more round. Here we go. Excitation, emission, and stop. And the arrival time is recorded. Okay. Yeah, but actually our TC-SPC electronics do more. They not just record the start-stop time of each photon with respect to the laser pulse, but also the time since the start of the whole experiment until the arrival time of the photon. And this will come in handy later. So yesterday I talked about how we can use this information to construct fluorescence lifetime images. Yeah, so where the fluorescence lifetime is measured in each pixel of the image. Today, we're not looking at imaging. Today, we would just park our excitation laser focus somewhere in the sample. So we'll just keep the excitation laser stationary, and then we have our molecule of interest diffusing freely inside the sample. Yeah, so this could be, for example, a labeled protein in solution. It could be labeled DNA or RNA in solution. Yeah, and so these labeled molecules, they are diffusing around in the water, for example, and whenever they come into the excitation laser focus, they start to emit a fluorescent signal, and then at one point they leave the excitation laser focus and no signal anymore. So if we now record the signal over time, and here we're talking now about seconds, yeah, this is the intensity time trace that we will get. Yeah, so here you can see there is a molecule, labeled molecule swimming through our laser focus, so we get a fluctuating intensity. So then the molecule is leaving the focus, so we get nothing, just background. There, a molecule enters the focus for a short time, going out again. Here's another molecule inside the laser focus for some time. So this is the kind of raw data that you would record. Yeah, and now there is actually the different different information contained in this kind of time trace. Yeah, so one is the intensity. Yeah, the intensity here can tell you about the number of molecules that you have in your focus. So this would tell you something about the concentration of the molecules in your sample. Yeah. You have the lifetime. Yeah, because as I said, we measure the arrival time of each photon.
Oh,
yeah?
Maria, sorry.
Yes.
Sorry to interrupt you. There is a problem with your microphone, maybe, because we're not listening to you, or I don't listen to you. I don't know if anybody have the same problem.
Okay.
You are okay now.
Oh,
Yeah, I'm listening. Okay, Ezequiel.
me too.
Okay.
Oh.
I'm just reconnecting. Is it better now?
Yeah. I can hear you now. Yeah.
Okay. Well, if that's better for you, then I'll just leave this. But let me know if the audio quality gets better again. Okay, so one dimension of information, let's say, is the intensity that we can look at, which would tell us, for example, about the concentration of the labeled molecules in the sample. But we also recorded the fluorescence lifetime on the scale of nanoseconds. Yeah, so we also have this information contained in our intensity time trace. We can also analyze these kind of time traces for FRET efficiency. And this is what I will tell you more about in a minute. And we could even look at anisotropy. Yeah, so I told you about anisotropy yesterday. For those who didn't join, so the anisotropy means we're looking at the polarization of the fluorescent signal, and this polarization orientation can tell us something about the rotational diffusion of the molecules. Yeah, so it could tell us how the molecules are rotating while they're moving through the laser focus. Yeah, we can look at this for images, but we could also look at this for time traces like these. Okay, so as I said, the system is recording across all the different time scales. Yeah, so from the nanoseconds, from the lifetime, across microseconds. So that's what I will look at in the second part of the webinar, all the way to seconds, where we can analyze these bursts of individual molecules going through the focus. Okay. So, and as I said, one very interesting process that we can look at in this way is FRET, first resonance energy transfer. Yeah, so short reminder, this is a process that takes place when two fluorophores come into very close proximity to each other. So one is called the donor fluorophore and one is the acceptor fluorophore. Now, if the donor fluorophore is on its own, then it's excited, and after a short time, it will emit a fluorescent signal. Yeah, nothing extraordinary. But now, if the acceptor molecule comes very close, then the donor is excited, but it will transfer its energy to the acceptor, and the acceptor is then emitting the signal. And this process is very sensitive to the distance between the two molecules. And this is in the order of 1 to 10 nanometers. Yeah. So this energy transfer can only happen if these two molecules are closer than 10 nanometers. So that means the labeled molecules really have to be directly next to each other. So they have to be interacting, basically. So we can also look at this again shortly in the Jablonski diagram. Yeah, so the donor molecule absorbs a photon, goes into the excited state, and then this excitation energy is transferred to the acceptor. Yeah, so there is no photon in between. Yeah, so this is a radiationless energy transfer, and then the acceptor is emitting this signal photon. Yeah. Also, this is taking place in the scale of nanoseconds. Yeah, so like the fluorescence lifetime. Yeah, and also this means that the lifetime of the donor molecule is actually changing. Yeah, so as said in the beginning, the donor molecule has a certain characteristic lifetime. Yeah, so it's excited, and after a certain characteristic time of a few nanoseconds, it will relax to the ground state and emit a photon. But now the donor molecule actually has two different options. It can go back to the ground state, or it can transfer the energy to the acceptor and thus relax. Yeah, so that means in case of FRET, actually the fluorescence lifetime of the donor molecule is shorter. And the more efficient the FRET process, the shorter the lifetime of the donor fluorophore will get. Yeah, so in this case, the fluorescence lifetime can tell us about the efficiency of the FRET process. So what can you use this for, this information of if two fluorophores are close to each other or not? Now, actually, with this information, you can get very creative. Yeah. So let's say you're looking at enzyme activity. Yeah. So you could, for example, label your substrate with donor and acceptor. And then if a protease is cleaving the substrate, then the FRET signal is lost because the two fluorophores are separated. Yeah. Or you could, for example, construct a sensor. Yeah, and so if the analyte is binding and the sensor is closing, then you have a FRET signal that reports analyte binding. Yeah. Another thing you could look at is conformational changes. For example, if you are interested in protein folding, yeah, so if the protein is unfolded, the two fluorophores are far away. If the protein folds, they come into close proximity, and you have suddenly a FRET signal. And very similarly, you could look at interactions of proteins, for example. So once the proteins interact, the labels come close to each other, and then you have a FRET signal. Yeah, so there are many different ways you can use this distance information. You can get really creative here, corresponding to your biological question. Mm-hmm. And actually, there's one field of research where single molecule FRET has proven to be really, really beneficial or advantageous. And this is dynamic structural biology. So what do I mean by that? Yeah, structural biology is studying the protein structure and how protein structure relates to the function of the protein. And there are, for example, many biochemical assays you can use to study this. And also there are other methods like X-ray crystallography, NMR, cryo-EM, which can be used to determine the structure of a protein. And that can even give you snapshots of different conformational states, which can give you a hint about how these relate to the function. But there are problems with these established methods. For example, some proteins don't crystallize, membrane proteins, for example. Other proteins are intrinsically disordered, so they don't even have a defined structure which you could try to determine. And then there's proteins that are, or processes that are intrinsically dynamic, such as protein folding or RNA folding. Yeah? So all of these are cases where a dynamic method like single molecule FRET can reveal information that you wouldn't get with these other methods. Yeah. And another big advantage is that single molecule FRET is working in solution at room temperature. So these are more, let's say, biologically relevant conditions than, for example, in a crystal or flash frozen. So this can, give you additional information that can, close gaps that you have with these other methods like X-ray crystallography. Yeah, and so I want to give you two examples to illustrate what I mean by this. Yeah, so the first example, we're looking at protein folding, and here this is the two-domain protein YPGK. Yeah, and the challenges that these researchers faced was to understand the folding of multi-domain proteins. Yeah, so the folding of single domain small proteins is pretty well studied and pretty well understood by now. But how about big multi-domain proteins? Yeah. So for example, here this YPGK has an N domain linker and a C domain. Yeah. And now what the researchers did is they labeled different residues in both domains and in between here. And then use single molecule FRET to measure the distances between these sites of the protein. Yeah, and then these could give them information about the structural changes during folding and unfolding, and complemented with molecular dynamics simulations, these can really give you a more complete picture of how the folding behaves. Yeah. So let's take a look at the data that they got. Yeah. So let's start up here. Yeah. We're looking at the N-domain mutant. Yeah. And here you can see the donor and the acceptor labeled here. And now they increased the salt concentration in the solution, and in the beginning the protein is folded, yeah, and so here you see a peak of the Fred efficiency corresponding to the folded state. And then here, if you turn up the salt concentration, you see that there is actually now two peaks appearing. Yeah, one is up here, so that's the proteins that are still folded, and then there's one here at an even higher Fred efficiency, which is a compact folding intermediate. Yeah, so if the folding intermediate is more compact than the original state, then the labels are closer to each other, so you actually get a higher Fred signal than before. Yeah, and afterwards, if you increase the salt concentration even more, then the folding at the protein is completely unfolded, so you have a low Fred efficiency because the two labels are far away from each other. Yeah, so in this case, the Fred efficiency can show you this folding intermediate before it completely unfolds. So you see that the interdomain mutant here is painting a pretty similar picture where you have a folded state with a high Fred efficiency. Then you have this folding intermediate somewhere here, and then you have this completely unfolded state with a low Fred efficiency. Yeah. And interestingly, now if we look at the C domain, this is behaving completely different. Yeah. So if we look at these two sites in the C domain, actually the Fred efficiency doesn't seem to be changing at all. And this could indicate a coil global transition. Yeah. So that the protein structure is changing, but without big volume changes, so that the distance between these two residues actually stays pretty much the same. Yeah, and so in this way, you can get a more, let's say, complex picture of the unfolding behavior of this protein. And this can be complemented very nicely with molecular dynamics simulations to understand what is going on here. Okay, and now I want to change gears and go to a second example. Now, in this case, we are looking at a folded protein that's ubiquitin. Yeah, ubiquitin is a signaling protein, and it can actually also be phosphorylated. Yeah, and now the researchers asked, how does the phosphorylation affect the ubiquitin signaling? Yeah, and it turns out that once you phosphorylate ubiquitin, it actually has two different conformational states that are shown here. One in blue and one in green, and you can see that they are slightly different, especially up here in this loop. Yeah, so the phosphorylated ubiquitin can have two states. One is the protonated relaxed state, and one is the deprotonated retracted state. Yeah, referring to this residue. Yeah, and also these two ubiquitins phosphorylated, they actually form a D-ubiquitin. Yeah, and this D-ubiquitin now they can actually have three interconverting conformational states. Yeah, so they can both be relaxed, they can both be retracted, or one can be relaxed, one can be retracted. Mm-hmm. And now if you label these two proteins, then the Fred efficiency can tell you which conformational state you are in. Yeah. So here the Fred between these two labels. Yeah, you can see if this is in the relaxed state. Yeah, you have a shorter distance corresponding to higher Fred. So if this is in the retracted state, the distance becomes larger, corresponding to a lower Fred efficiency. Yeah, so in this case, you can use single molecule thread to visualize the conformational distribution here between these proteins. Yeah, so you can, this is the data that you get. And you can fit this with 3 peaks corresponding to 3 different thread populations. And now you can compare the population of each state in different conditions. Yeah, so in this case, they changed the pH. Yeah, and then the change of pH actually changed the conformational distribution. And this is, oh, I just see there's a small mistake here. That should be a different pH value, sorry. But this means that the variation of the pH actually changes the dynamic equilibrium of these structures. And this would then in turn change the signaling behavior of this d-ubiquitin. And this is then very interesting because the pH varies among organelles. So that means in different organelles, you could actually have different signaling behavior for the same signaling protein. Okay. So with this, I want to take a short break for questions before changing topic and moving on to the FCS method. So are there any questions?
Yes, I have a question. I understand that this is done with a very dilute solution, so you get the situation of a single molecule, right?
Or a few molecules, yes. So
A few molecules. So,
yeah.
What is the time that actually one single molecule is bombarded with these photons? I mean, and how? And the amount of photons that will actually look at that single molecule.
Well, that depends a lot on the diffusion coefficient of your molecule in the solution. So,
but you get enough data in order to
yeah. So, so usually yes. So the molecule, how long it stays in the laser focus depends on the diffusion coefficient. Yeah, so on the size of your protein, the viscosity, and so on. But yes, usually you get enough data. You can watch it diffuse through the focus, maybe even come back again. Yeah.
So if you get another molecule supposedly identical, you should get about the same data.
Yeah. Yeah, you could see this here, for example, in the time trace. Yeah, you see each burst is approximately the same number of photons. Yeah. So I couldn't even say if this is molecule A and this is molecule B and molecule C, or if this is molecule A coming shortly back and this is molecule B.
Oh,
so
you cannot tell. The data
I can't tell in this case, no, because it's all this, let's say it's the same protein labeled with the same GFP, for example. Yeah, so I can't, you couldn't tell them apart.
Okay, thank you very much.
Mm-hmm. Yeah, so this works with statistics. Yeah, so you look at a lot of these bursts, yeah, at a lot of these peaks, yeah, and then you make statistics to see, for example, that here, yeah, here, for example, you see, okay, 68% of these peaks have high Fred, 23% have intermediate Fred, 9% of these peaks have low Fred. Yeah, so I don't know which peak is which, but yeah, so these, you look at the statistics here. Does that answer your question?
Yes, certainly.
Yeah.
I have one question also. For example, the amount of photons that you get from your sample obviously depends on its diffusion time, for example, because as you said, the molecule is gonna move more or less time depending on its diffusion coefficient. But Also, the amount of photons that we get from the experiment depends on the repetition time of the excitation, right?
Mhm. Yeah, so on the excitation intensity, yeah, so you could turn the intensity up until you start to see bleaching of the signal, then you should turn it down again. Yeah. Also the intrinsic brightness of your marker, yeah, so some marker are brighter than others. Yeah.
But for example, if we have our repetition rate of excitation, for example, I don't know, I'd work with 40 megahertz repetition rate for excitation. If we change, for example, 10 or 20 or even 80, I think it's for multi-photon excitation, right? But
yes,
how that repetition rate affects the observations that we can finally get from our experiment.
So if you, let's say you double the repetition rate, so from 20 to 40 megahertz, you get twice as many laser pulses, so you would double the intensity of your signal.
Oh, okay. So you can get more short experiments or yeah.
You would just have to see if the fluorescence lifetime of your molecule still fits into the, the time between the laser pulses. Yeah, so if you have 80 megahertz repetition rate, that means you have 12.5 nanoseconds between pulses. Now, if you have a short lifetime of 2-3 nanoseconds, sure, this will decay completely before the next pulse comes.
Exactly. Okay.
If you have a long lifetime of like 5 nanoseconds, then it can happen that the signal doesn't decay completely.
Yeah, so you can
next pulse.
Yeah, you should decrease the repetition rate in that case.
Yeah, so if you want to look at the lifetime, if you want to analyze the lifetime, that's something you should take care of. Yeah, but the good thing is with our diode lasers, you can freely choose the repetition rate that you want to use. Yeah, so you can choose the value that is optimal for the lifetime of your marker that you're using.
Another thing that I want to mention in these experiments, because I work with this, is the capability to observe not only the donor lifetime but also the acceptor lifetime. If you have, for example, detectors for obviously both photophores. So in that case, another component interesting to look up when you are making this kind of experiment is see the stoichiometry of your samples to observe when, for example, your acceptor is photobleached or when your acceptor is absent. Also, you can directly relate your FRET signal to this other component to see if you have, for example, low FRET signal because obviously your photophores are far away from each other, or if you have no FRET or very, very low FRET because you don't have an acceptor or your amount of acceptor is decreased because it's photobleached, right?
Yeah, that's a very good point. Absolutely. So if you look at the acceptor signal, it's a very good internal control for bleaching and things like that. Yeah.
Yeah, we can do that in our equipment because we have laser for donor and acceptor, and obviously we have the detectors for the donor and acceptor. So this is a very good control for all kind of experiment related with FRET in this upgrade of the Pico one.
Yeah, that's a very good point. Thanks.
Yeah. It seems like there is no question in the chat. No, there is no question. Okay.
Yeah. Regarding a single molecule approach to study protein flexibility, if I label, let's say, a loop of a protein with one fluorophore, is it possible to measure if this region is more flexible than maybe another region I had labeled in another sample? Not maybe to FRET, because that will require FRET resonance, but maybe to anisotropy. If with one region label I have more anisotropy, if it is a more flexible region, is it possible to make that kind of analysis?
That's a good question. So, okay, what anisotropy is telling you is basically about the orientation of your fluorophore, your marker molecule, and how this changes on a short time. But the question is, does it change because the entire molecule is rotating, or does it change because your, let's say, your loop is flexible and moving around with respect to the rest of the protein? That's the question for me, would be how to tell this apart, how to know.
That part of the question that you mentioned is very important for us also. We have the same question when we are making our experiment. Usually for Fred, you want your fluorophore very freely diffusion, yes. Because any change in the position of the fluorophore on the conformation or of the fluorophore can affect the Fick signal. Mm-hmm. But for anisotropy, that property of the fluorophore is not good to understand the question that Fick has, because as you mentioned, the property or Alexa, for example, which has a very long linker, and then the fluorophore is freely diffusion, moving around the protein. And then if you look at the anisotropy of your sample, you are going to be a very large fraction of anisotropy component regarding the movement of the fluorophore. And then another fraction of the anisotropy component would be the global component, assuming the fluorophore and the protein. But for example, if you change the fluorophore to another fluorophore that is more tightly linked to the protein and more close to the chain, the anisotropy component is going to be changed, because the major component, it would be related with the chain, because the fluorophore is very tightly linked to the protein and very close to the protein chain, and the freely diffusion component of the fluorophore decreases in that kind of experiment. For example, we use body pi fluorophore, which is a very short link that connects to the protein, and then the anisotropy component is very related with the movement of the chain, where the fluorophore is linked. So in that cases, I could say that the anisotropy is not the main technique that allows you to say how the dynamics is the protein or some parts of the protein, but maybe tells you about possible changes of the anisotropy component of that part, specifically of the protein, whatever you changes the environmental conditions or the presence of some ligand, for example, if you have an enzyme or whatever. So then the changes in anisotropy tells you about the possible changes in that region of the protein, very close to the fluorophore in this case, but now tells you to match, at least. We are working on that now in our recent work, but the answer to Sixto could be that it depends of the fluorophore that you use for examining the anisotropy. But obviously the most elegant, informative way to understand dynamics in proteins is making Fred experiments.
Mm-hmm. Yeah, definitely very, very good point. But then I guess it couldn't hurt, if you look at the same sample on the same instrument, but with different methods. Yeah, so you could do anisotropy experiments, you could do Fred experiments, perhaps you could do FCS experiments, and these would look at the same sample from different angles. Yeah, so you perhaps if you combine all this information, you could get a more complete understanding. Yeah, and if let's say the anisotropy and the single molecule Fred all point in the same direction, then you could be more confident that you're understanding the behavior of your protein correctly.
I clear now.
Okay,
I think we can change the topic now.
Mm-hmm. Okay. Yeah. So we've been talking about the nanosecond region. That's the fluorescence lifetime decay. We've been talking about the second time scale where we're looking at these bursts of single molecules. Now I want to go in between to the microsecond time scale to look at the correlation of the fluorescence signal with fluorescence correlation spectroscopy, FCS. Yeah. So what's happening? Yeah. Again, we are parking our laser spot somewhere in the sample, and we're watching single molecules or few molecules diffuse through the laser focus. Yeah, and this causes a fluctuation in our fluorescence signal. Yeah, and so here we are recording the fluorescence intensity over time. Yeah, and here you can see, well, the mean intensity already tells you about the number of molecules that you have in your focus on average. Yeah, and if you have few molecules, you have less intensity, more molecules, higher intensity. And you can also look at the intensity fluctuations over time. Yeah, and you can imagine, for example, if you have the difference between 1 and 2 molecules would be very large fluctuations. The difference between 10 and 11 molecules would already be fewer fluctuations. Yeah, so what we are looking at here now is the intensity fluctuations in this kind of time trace. Yeah, and now there's the software is doing some mathematics that is called the autocorrelation of this signal. Yeah, so the autocorrelation of the intensity changes over time, and these result in these correlation curves. Yeah, and these correlation curves look like this. Yeah, where the amplitude here is telling us about the number of the molecules. Yeah, so it's 1 over the number of the molecules in the focus. This means that the amplitude is telling us about the concentration of the molecules in our sample, and this is absolute concentrations. Yeah, whereas on this axis, this is the lag time. Yeah, so the characteristic correlation time. And this is telling me about the diffusion behavior, yeah, the diffusion coefficient of the molecules as they move through the laser focus. Yeah, so if I have a longer lag time, it means a slower diffusion of the molecule through my laser focus. Yeah, so these are the two basic pieces of information that I can get from such a correlation curve: is the diffusion coefficient of my molecules and is the concentration of the labeled molecules in my sample. Yeah. Which seem like, let's say, pretty basic, but there is a lot of interesting conclusions you can draw from this. Yeah. So one, for example, is looking at the expression levels, yeah, in living cells. Yeah. So you could use concentrations, for example, to determine expression levels. Yeah. Another one is looking at the diffusion. This could tell you, depending on your sample, it could tell you about protein folding, for example. Yeah, so a tightly folded protein will diffuse differently from a large unfolded protein. Yeah, or if you're looking not at proteins but at, for example, membrane proteins or at labeled lipids, this could tell us something about the behavior of lipids, for example, in a membrane. Yeah, how they diffuse in the lipid dynamics. Yeah. And we could even use FCS to look at molecular interactions, for example, complex formation or binding, unbinding of proteins. Yeah, because you can imagine that if you have 2 individual proteins, they will diffuse faster, independently of each other. But if they bind together and you have one large complex, this will diffuse much slower. Yeah, so this is also something you can look at. Yeah. And now I want to give you two examples how you could use this. Yeah, one is here. We are looking at G proteins in so at the human cone opsins. These are inside the human eye. Yeah, that allow us to see color. Yeah, so we have red, green, and blue opsin, which can allow us to see red, green, and blue color, color vision. And the question was, are these opsins in actually, when they're in the membrane, are they monomer or are they a dimer? And so the researchers now expressed these opsins in the cells, parked the laser focus on the membrane, and then watched the opsins diffuse through the laser focus, and they recorded this kind of intensity time trace. And then by correlating the intensity time trace, I got this correlation curve. And here again, the amplitude is telling us about the brightness, and the lag time is telling us about the diffusion coefficient. And so they could see on the one hand that the red opsin appeared to be much brighter than the green and blue opsin. And on the other hand, the red opsin is diffusing much slower than the green and blue opsin. And taken together, they concluded that actually the red cone opsin is a dimer in the membrane, whereas the green and the blue opsin are monomers in the membrane. And now for a second example, we're also looking at membranes, but now we are looking at prion proteins. So prion proteins are proteins that misfold, and when they misfold, they aggregate into particles, and these particles cause prion diseases. And it's known that the native prion protein, so in the healthy state, is tethered to neuronal plasma membranes. That's why these researchers asked, what's the role of the membrane properties in the misfolding of this prion protein? So they assessed the diffusion of this membrane protein under different conditions. So for example, in the normal non-treated membrane, and then they did different things like, for example, depolymerize actin to see or remove cholesterol from the membrane, and to see what these different conditions would do to the diffusion properties of this prion protein. And for example, here they looked at the diffusion of the labeled prion protein and saw that if they add LAT-A, which disrupts the cytoskeleton, then suddenly the prion protein is diffusing more slowly than in the non-treated condition. So apparently the cytoskeleton is one of the factors that affect the behavior of this prion protein. So you can see that the diffusion coefficient, you measure the diffusion coefficient itself, but if you measure it in different biological conditions, this can reveal interesting aspects about your biological system under study. And here one last example where we're also using FCS, but in this case to look at phase separation and how the aromatic residues in these prion-like protein domains determine the phase separation behavior of these proteins. So here the idea is that you have these intrinsically disordered prion-like proteins, and that these could, stick together, form these droplets, by liquid-liquid phase separation. And then you can use FCS, or you can do FCS in different places in the sample, so inside the droplet and outside the droplet, and then you can even compare the diffusion behavior inside and outside droplets to get an idea of what the behavior of the protein in these different microenvironments is. And if you combine this with, for example, computer simulations, this can give you an idea how the let's say, how the dynamics of your protein correspond to the phase separation behavior. Yeah. Okay. And so here the diffusion coefficient from FCS can, you can relate it, for example, to your computer simulations to confirm if the if the picture you're drawing is actually plausible. Okay, and with this, I'm actually at the end already. Yeah. So I told you on the one hand about analyzing these intensity time traces for single molecule FRET to read out, for example, FRET-based biosensors, or to monitor protein-protein interactions, or to look at conformational subpopulations of proteins. And on the other hand, I told you about fluorescence correlation spectroscopy on these intensity time traces to measure diffusion coefficients, which can tell you a lot about, for example, protein complex formation or phase separation behavior, and also about determining molecular concentrations in your sample. Yeah, and with this, I'm in the end, and I'm happy to take any questions either now or if questions come up later. You can always email my colleagues and me, and you can also try and check our time resolved wiki if you can perhaps find this topic there as well. And of course, thank you for taking the time, and thanks for your attention.
Okay, thank you, Maria. Very nice presentation. I want to add to the last part of the presentation that we actually can, in our group, combine very nicely the FCS and FRET information to make that we call the FRET-FCS analysis of the sample. So with that kind of combination, you can actually get dynamics from the nanoseconds to milliseconds in our experiment analyzing the protein dynamics. So it's a very powerful mode to analyze protein transition on characterize disorder, characterize the unfolding processes. So combining this entire presentation allows us to get very useful and high resolution information in terms of structure and in terms of temporal changes. So very nice presentation. I thank you to give us these two presentations, the yesterday presentation regarding FLIM and the today's presentation regarding more, let's say, structural biology, like the mode to understand these methodologies. But I think were both very useful to understand a little bit, at least more, what kind of things, what kind of information we can extract from our experiment, so that way we can think maybe another level, another layer of information about our biological question, as you mentioned in the first part of the presentation.
Mm-hmm.
So.
And as you said, it's great if you can even combine the different methods, yes, to look at your same biological question from different angles, yeah, to see
exactly
if all the pieces of the puzzle match with
exactly
each other.
Exactly. So we now can, for example, make a biological big question. For example, what is the relevance of, for example, the increasing of disorder on some protein, of the effect of some post-translational modifications in a protein, in, for example, the binding to ligand to phase separate that you mentioned in the last part of the presentation. So we can understand, for example, observing the protein itself, and then change the observation to the cells and use FLIM to understand that part of the question and combine all together and see a beautiful main picture about how the relationship between structured and unstructured proteins and function can can be combined and related in order to understand how that affects, for example, a cellular process. So I think these two presentations were very informative in that way, in terms of how we can combine up these methodologies to make more bigger questions, or the same question but answer it with more resolutive approximations.
Mhm.
So thanks for your presentation again. These two presentations or these two webinars are already available in my YouTube channel. So if you can't get to my YouTube channel because I'm not famous, you can also contact me via email and send the YouTube link. So actually, you can revisit anytime these presentations are. Understand a little bit more about this stuff. I don't know if Dr. Babul have to say
Yes,
something.
thank you very much, Maria. Very helpful, and we're very glad to have this equipment, and a lot of people will be using it, and we'll keep in touch and telling you of our efforts in order to advance in this new area at least for our country. Thank you very much. And maybe, next time it will be a new instrument. I don't know how you're developing these things.
Yeah, I'm also really looking forward to see what what new research you will do with this experiment. So I'm very excited to read all about it in a year or two.
Of course. Thank you. We'll see you then. Bye now.
As I mentioned in the first part of the presentation, we will have additional webinars from actual investigations that are running in different areas using FLIM, PrEP, FCS, single molecule, whatever. And then all you guys can think more in the research or see the research component of these methodologies, and not just the background information, but these two presentations, very good basis to start to think about how with these methodologies we can answer some questions. So maybe if you have questions about this presentation, you can contact me or contact Maria. So we're going to be glad in answer your questions.
Yeah.
So
more technical and practical aspects, grab Volker, my colleague Volker, when he's visiting you, because as I mentioned yesterday, he has years and years and years of experience of actually doing this kind of experiments. Optimizing parameters, samples, and so on. Hi, Volker, there he is.
Hi, Volker.
Yeah. So yeah, he can. I think he will be happy to share all of this with you as well.
Yeah, we'll see Volker in a couple of weeks here. So we can talk about the practical methodological aspect of the system that we installed in our faculty. So I hope to see you soon, Maria. I'll see you soon, Volker, to talk about this system and the installation. That's it. I don't know if you have some additional comments to say, or we are okay. Maybe we're okay.
From my side, nothing. If any questions, anything comes up, feel free to email me or call me. Yeah, and otherwise, yeah. Okay. I already wish you best of luck with the system.
Okay.
Thank you.
Okay, so thank you all of you guys for your presence, your attendance, and we'll see you soon, Volker. I hope to see you soon, Maria, to talk about this system. And thank you again for giving us these nice presentations.
My pleasure.
So we are finishing